Neurons. What are they and how do they work? The function of neurons. Neurons are unique cells designed exclusively to transmit information throughout the body. They possess specific characteristics to make them highly effective at carrying electrical impulses known as action potentials. They have dendrites, which receive signals from neighboring neurons, axons, which transmit signals over a distance. These may be covered in myelin sheaths, which speed up transmission signals along the axon. And they have axon terminals, which transmit signals to other neuron dendrites or to tissue. Axons and dendrites, when they meet, they do not touch. There is a space between them, known as a synapse. Understanding action potentials and their role in signaling within the body. Electrical impulses, called action potentials, facilitate communication within the body. Prior to an action potential, when the body is in a resting state, all gated sodium and potassium channels remain closed. Understanding the resting membrane potential of neurons. Neurons tend to have negative concentration gradient, which indicates that there are more positively charged ions outside of the cell than inside. This state is referred to as the resting membrane potential. Here's what happens during that phase. There are more sodium ions outside than inside the neuron, and there are more potassium ions inside than outside the neuron. There are 5 main events that take place during an action potential. First, triggering events and neurotransmitters impact ion flow. Positive ions continue to flow into the cell, yet they can only pass through channels that are open. This happens when a specific chemical, called a neurotransmitter, binds to the channel and signals it to open. Neurotransmitters are typically released by cells near the dendrites, often at the conclusion of their own action potential. Once that triggering event or stimulus occurs, and the charge of the cell passes the threshold of excitation, then depolarization of the cell will occur. Depolarization of the cell is the process by which a cell becomes less polar. It occurs when positively charged sodium ions flow into the negatively charged axon, causing the surrounding areas to depolarize. This is similar to a domino effect. Once one channel opens and lets positive ions in, it triggers the channel down the axon to do the same. The maximum action potential is reached once the signal travels to the end of an axon terminal. This causes the release of a neurotransmitter, which will then trigger an action potential to occur in another neuron. During repolarization, the cell returns to its resting state by following a specific process. Firstly, the gates of the sodium channels close, which halts the influx of positive ions. Simultaneously, the potassium channels open and the potassium inside the cell exits. As a result, the cell loses positively charged ions and returns to its resting state. Hyperpolarization is a process that makes the cell more negative than its usual resting state. During an action potential, potassium channels remain open slightly longer, allowing positive ions to exit the neuron. This results in temporary hyperpolarization. The potassium channels close and the sodium-potassium pump activates to restore it to its resting potential. How drugs affect the brain's neurons and neurotransmitters. Drugs can disrupt the way neurons transmit, receive, and process signals through neurotransmitters. Some substances, like heroin or marijuana, have a similar chemical structure to natural neurotransmitters, allowing them to bind and activate neurons. These are called agonists. However, they don't activate neurons in the same way as natural neurotransmitters, leading to irregular messages being sent throughout the network. Other drugs, such as cocaine and amphetamines, disturb neuron communication by either releasing abnormally large amounts of natural neurotransmitters or preventing normal neurotransmitter recycling by interfering with the transporters. The actions of these drugs, called antagonists, distort or amplify the normal communication between neurons. Let's see this in action. So here we have a normal neurotransmitter, and the signal has been sent. But what if we have an agonist that comes in and disrupts? The agonist causes a different signal to occur. As you can see, it was a bit different. This would lead to irregular messages being sent throughout the network. What about antagonists? Well, here again, we have our normal neurotransmitter coming in and sending a signal. But what if we have an antagonist that comes in instead? The antagonist takes the spot and will either send abnormally large amounts of neurotransmitters to be released, or it will come in and prevent normal neurotransmitter recycling by interfering with the transporter and blocking the neurotransmitter from entering. All right. Well, I hope you've learned something here and can do well on your exam. Good luck.